So I'll tell you a little bit about, so you got a really nice introduction about the CRISPR and editing technologies, and I'll tell you a little bit how we are using these technologies in order to do genome scale, genome-wide modifications to study a lot of genetic elements in parallel. So you all, a quick recap. So we basically harness, or we take advantage of the repair mechanisms and to introduce a double-strand break. And everything I'm going to tell you now is basically based on this side of this figure. We use targeted double-stranded break in order to introduce mutations into early constitutive exons. And what happens here is that because the non-homology repair is error-prone, then you get an indel mutation that introduces a frame shift. A frame shift usually will introduce at some point a premature stop codon, and then this gene will not be active due to all sorts of NMD and quality control mechanisms of the nucleus. Okay, so the previous generations of these, you heard about them, and then came CRISPR at 2013. And really one of the main there's a few advantages: the ability to do multiplexing, the very high efficiency of modification. And in our case, one of the most appealing things was really that target specificity is encoded in the short RNA molecule and not in the protein structure like in zinc fingers and talents. Okay, so if I want to switch the loci and talents from one location in the genome to another, I need to engineer a new protein. But if I do it, but with CRISPR, to do it, all I need to do is synthesize a new sgRNA, or in this case, because we put everything in a plasmid, you just need to synthesize a short oligo, clone it in the plasmid, and you have a new plasmid that targets a different location in the genome. And that really opens the possibility to do a genome-wide modification. So when we usually think about genome editing, the first thing that comes in mind is really I have some genetic element of interest and I would like to perturb it in cells and then explore multiple phenotypes, growth, transcription, any phenotype that you're using, and see basically what does it does. But the opposite error in this is basically, given a specific phenotype, can I use these technologies in order to find all the possible, or basically screen through thousands of possible genetic modifications in order to find additional genetic modification that actually give rise or change this specific phenotype. And of course, this is not a very new concept. So people have been using shRNA screens and overexpression screens in order to do these types of studies. And the first thing that we did basically is to, together with my collaborator, Neville from the lab, was to see if we can use CRISPR in order to do this. So the first step was basically to how do we deliver CRISPR in such a pooled fashion. So what is traditionally done for shRNAs is to use lentiviruses. So why do we use lentiviruses? First of all, we get constitutive expression of Cas9. Lentiviruses integrate into the genome, so we can basically read out which sgRNA was in which cell at the end of an experiment. And also, it's pretty established to control the multiplicity of infection with lentiviruses. So you can very easily introduce one virus to one cell, which is very important for pooled screens. So we engineered this plasmid that contains U6 sgRNAs and Cas9 in a lentiviral backbone. And one of the main concerns here is that before this stage, what everybody was used to deliver Cas9 were actually transient transfections of DNA. And in this case, what you do is you introduce into the cells a lot of DNA, and this DNA gets dilutes out of the cells pretty quickly. Okay? And the question was whether a single virus would be good enough to have high enough efficiency of genomic modification. So to test this, what we did is we took this construct, we made virus, and we introduced different sgRNAs to target an EGFP that is integrated into these cells. And what you see here is basically targeting of this EGFP. These are histograms of fluorescence. This is the wild type cell that just expresses GFP. And this is after 10 days of selection. So you see that most of the cells, this is targeting with Cas9, and you see that most of the cells are completely indistinguishable from cells that do not have the gray color here indicates cells that do not have VEGFP. So it's basically almost 100% genomic modification. This is as comparison shRNAs, and as expected, shRNAs, they only reduce the expression of the protein. So you see that the whole distribution is shifted, but it's not completely knocked out. One thing interesting to observe here is that in some cases you see a bimodal distribution, right? Why would you see a bimodal distribution? Because what we know to do with Cas9 is to introduce a mutation, an indel. And whether or not this indel is going to actually cause a disruption of the protein function, that's what happens. Sorry. I agree. All right. That's a question that's specifically to the gene and to the exact location that I actually introduced this indel. So we also tested the genomic modification, and indeed we find very, very high allele frequency, modified allele frequency. Okay, so we reach basically almost 100% of modification, and that's very good for screening. Interestingly, when we look so one of the concerns is basically, okay, I'm expressing Cas9 for a long time, I'm increasing my on-target efficacy, but I'm also increasing off-target. And surprisingly, we didn't do a very, very extensive test, but for quite a few genes that we tested, and this is the on-target modification rate. This is after 7 days. This is after 14 days. And at least in the predicted off-target that we know how to predict them, we don't see very high levels of off-target modification. In some cases we do see, but in most cases we don't. Okay. So basically, we engineered two delivery systems. One delivery system in which we deliver Cas9 and sgRNAs together in one vector system, and everything that I'm going to show you is basically based on this delivery system. We also engineered this delivery system in which we first introduce Cas9, expand the cells, and then introduce sgRNAs. This is available. But everything that I'll tell you is actually based on this system right now. So the experimental outline, you know what, let's skip this. Let's go straight here. So what was the first step in order to introduce, to make a genome-wide knockout library? So we used the information that was generated by Patrick and other people in the lab about off-targets, and basically devised some computational model in order to calculate for a given sgRNA an off-target score. So we go through all the genome. For each gene we use splicing data in order to recognize early constitutive exons. For each one of those exons, we go through all the sgRNAs possible, and for each one of those, we calculate an off-target score based on the similarity across the whole genome. And the scores basically incorporates a few different parameters that were empirically observed in these studies. So we know that the position of the mutation will have an effect. We know that the density, whether the mutations are close together or spaced apart, will have an effect. We know that the number of mutations will have an effect. So we try to integrate all this knowledge, and this knowledge also gets updated all the time. So now we actually changed our formulas a little bit. Okay, so we designed a whole genome library. And the next stage is, because these are relatively short sequences of only 20 base pairs, then we can synthesize them on programmable microarrays. So these are basically like microarrays. You design your sequences on a computer, and then you make this microarrays with all the short oligos. These are all pooled together on a single chip. You can then chemically cleave those off of the array and basically get all these oligos in one specific in one tube. You then use these oligos to do a pooled cloning. So you take the synthesized oligos, you clone them into the vector that contains the sgRNA and Cas9 components, and you get a pool of plasmids. Okay, so in our case, we designed 65,000 different sgRNAs targeting more than 18,000 genes. After cloning, we get a plasmid pool. And after making virus, we get a viral pool. Every virus will target a different exon in the genome. We then transduce cells with this library. and select for only transduced cells. So when you do transduction in low multiplicity of infection, because you want to make sure that every cell gets a single virus with very high probability, then you will infect around 30% of your cells, but then you're going to select. So we basically fuse Cas9 to a puromycin selection. So you're going to select only transduced cells. So at the end of this process, you're going to end up with a pooled cell knockout library. Okay, you have a big pool, a big flask of millions of cells, and every cell has a specific mutation corresponding to a different gene knockout. Okay, so the first step to show, we wanted to show that we can use this system in order to perform both negative and positive selection screens in human cells. So negative selection screens, you're basically looking for gene knockouts that kill the cells or basically drop out from a specific assay. And positive selections are actually knockouts that are enriched in a specific assay. So we use two cell lines. One is a melanoma cell line, A375. I'll talk a little bit more about this cell line in a minute. And also HU62 human embryonic stem cells. So we infect these cell lines with a library. We transduce it with a library, select with puromycins, grow the cells, and basically look at the distribution of different sgRNAs. So one thing that I didn't say is that we basically use the integration of Cas9 into the genome as kind of a barcode. So because it's integrated, after 2 weeks, you can just take the cell pool, you selectively amplify the provirus integration, the lentivirus, and use the spacer as the barcode to basically count how many times I have from each sgRNAs in this specific pool. Okay? So we do this, we quantify, we basically count how many sgRNAs we have in the pool right after transduction, and how many sgRNAs we have from each, how many in the pool after 2 weeks of growing the cells. And as expected, so we see a reduction, so we see a shift in the distribution that corresponds to some sgRNAs which are actually are actively dropping out of the pool. And when we look for enrichment, we see that these are indeed very, very highly enriched in very essential categories like RNA processing, structural constitutive of the ribosomes, et cetera, et cetera. So this is basically for us that this was kind of a proof of concept that you can use this library or this approach in order to do negative selection screens. The next step, what we wanted to do is to show to you to do positive selection screens, which are actually much stronger. So we use the melanoma cell line model. So A375 cells have a mutant BRAF, V600E, and that's one of the most common mutations in melanoma, found in more than 70% of melanomas. And we used recently, not very recently, but like 2 years ago, approved drugs, PLX, vemurafenib. And what happens in patients here is basically, you can see here a horrific picture of melanoma before PLX. So after these people are treated, so this is a drug that is designed to target this specific mutant form of BRAF. Okay, so it specifically inhibits this specific mutation. So what happens in these patients is that there is remission. You see that the tumors actually go away. But pretty early on, after a couple of like 3-4 months, there is recurrence because these cells actually acquire mutations that enable them to avoid the therapy. Okay? So the question here was whether we can use this system in order to find novel mutations that confer resistance confer resistance of melanoma in this specifically model to this specific drug. Okay? Okay, so how does the experiment go? We basically take cells, we transduce them with a library. We call it GeCKO. There's been a very very controversial acronym. Some people really like it, some people really hate it. So we take these cells, we transduce with the library, we select for puromycin for transduced cells, and then we put on the drug. You see here that cells without the drug will continue to proliferate, and cells with the drug, they have a cytostatic effect, so it's basically stop proliferation. And this basically enables the resistant cells to enrich in the population. Okay. Okay, so now we basically, in different time points, day 0, day 7, day 14, we take the whole population of cells, we PCR amplify the provirus, we count how many sgRNAs we get basically it's how many cells we get from each sgRNA. And what you see here is that the distribution, this is basically shows you the distribution of representation of the different sgRNAs, and you see it, and over on the drug at day 14, you can see a very large shift in the distribution, such that most of the sgRNAs are depleted, but very few are actually very highly enriched, and this is a log scale. So this enrichment is pretty strong, and you can see also by just unbiased clustering that the day 14 is actually pretty different than the other samples. This is just to throw some data on you, so you'll believe better of what I'm saying. So here you see, for example, a comparison between two biological replicates of control at day 14, and you see that the sgRNA representation is pretty nicely correlated. But here, for example, if you compare, so PLX day 14 to, let's take it to the control day 14, then you see here the enrichment of a small group of sgRNAs, okay? Okay, so we looked into this cloud of points, and we were very happy to see that a lot of them were actually sgRNAs that target the same gene. Okay, so you see here NF1 was targeted, but 4 different sgRNAs for NF2, 2 for NF1, COL3. And the next step in these kind of experiments is to see if we can basically take so this cloud contains a lot of false positives and some true positives, and you need to pull out the true positives. So the way this is done is basically to use some algorithm, and there's a few algorithms out there to do that, that takes into account the level of enrichment and also the consistency of different reagents that target the same gene in order to find the true gene hits out of this big cloud of points. So you see here that once we use this, then here actually the background, so here in the background there's a lot of points. So you would tell me, oh, this is great, but what are these guys here? How come you have so many gray points that you don't highlight in your figure? But here you don't see a lot of them, right? Because we basically use some statistics in order to find the genes that have multiple sgRNAs that are actually enriched. And we were very happy to see this because we were actually expecting NF1 in this screen. So NF1 was previously implicated, loss of NF1 was previously implicated to give resistance in this specific cellular model. So when we found it, it was like, yay. As I woke up my wife at 2 a.m. when I saw this data. And we also found MED12, which was also previously shown to give resistance here. We also found NF2, and I didn't know a lot about NF2 at that point, but it has the same name like NF1, so it has to be related in some kind of way. Interestingly, it's actually not sequence related, so these two genes were given similar names because of the phenotype. They actually act through very distinct pathways, which is also very interesting for people who study melanoma. So this was good. This is just to show you that these experiments when you analyze these types of data, it's like a word of caution. So every experiment that you're going to do is going to have a huge, this is considered a low background, but still you have some background. So this is just an example of, and I also debate with myself of whether I want to put this slide in or I don't want to put this slide. But I'll put this slide here. So this is basically, these are 2 infection replicates, so complete biological replicates of the whole experiment. Every point here corresponds to a single sgRNA, and the axes are basically the level of enrichment, the level of fold enrichment between the control, the MSO, and the drug treated. And when you first look at this, it looks pretty horrible, right? Completely uncorrelated. And the reason for that is that most sgRNAs will have no effect. And they're just going to, most lentiviruses will have no effect, and they're just going to be used as kind of a cell barcodes, right? So, for example, if I kill 99% of my cells with a drug, I still have 1% that are going to survive either due to some stochastic things or due to additional acquired mutations that I don't actually measure. And so all of this big cloud is basically my background. But when you look at things that are actually reproduced between the two replicates, and you zoom in into these, you will see that all of these are actually actually all of them are actually sgRNAs that are similar sgRNAs that target the same gene. So you see here NF2, 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 NF1, NF2, col3. So basically, infection replicates and multiplicity of sgRNAs that target the same genes are the main tools that we used in order to pull out the true positive out of the cloud of false positives. Okay. The main thing that we wanted, so technologies for pool screening actually are available. People do it with shRNAs or with overexpression ORFs. And we wanted to do a head-to-head comparison with shRNAs. So you can think that shRNAs are incomplete knockdown. There might be some application in which you would actually want that. And this specific screen, the reason why we chose it is because some of our friends in the fourth floor actually did it with shRNAs. So what you see here is the top hits from an shRNA screen for PLX resistance in melanoma, and the top hits for the CRISPR screen that we did for the same cellular model with exactly the same parameters. And the bars here correspond to the percentage of sgRNAs that are actually enriched in like 5% of the population. So you see that the top hit, NF1, that we were actually expecting in shRNAs is still less than 50% of the shRNAs are actually enriched, and the other, the other hits are even, these were actually not validated, and they're much less. And you see that for the CRISPR, it's really promising. So this is not a small improvement. It's a really, really huge improvement. You see that in a lot of these cases, 100% of the sgRNAs are actually enriched. So all of them work, which is really, really great. We also did all sorts of statistical methods in order to quantify it. So there is a rigor algorithm to do this, and RSA. And in all cases, if you look at the cumulative p-value distribution, you see that GeCKO CRISPR is always lower than the p-values for the top hits for the shRNAs. So statistically, we're doing much, much, much better with CRISPR compared to shRNAs. Okay. So we wanted to look, first of all, we validated our hits. So I have to say that we tried to validate the 7 top hits in a RAID format, not in a pooled format. 6 out of them were actually validated. And that's a very, very high, for people that did previously shRNA screens, you would know that this is an extremely high validation rate. So specifically, we looked at NF2 specifically, and we basically asked ourselves, okay, why did this gene did not come up in the shRNA screen? Like, this is a very strong, in our screen, it's the top hit. And as you see here, this is the basic dose response curve for the CRISPRs targeting NF2 with different sgRNAs and under PLX and different amounts of PLX. And you see that the rescue is very, very strong. Like, the phenotypic effect is very strong. And how come we didn't find it with the shRNA? So if you do a Western blot of this protein using both sgRNAs and shRNAs, you actually see depletion of the protein also with shRNAs. But when you look at the phenotype and the phenotypic effect, you actually don't see a very strong effect with shRNAs. Only with one of them you see a small effect. Now, why is this? So you can hypothesize a little bit. What we basically think is that the distribution of even for good shRNAs, the distribution of expression, as I showed you at the beginning, It's basically going to shift the distribution quite a bit, but it's going to shift the distribution, right? You're still going to get a small amount of expression. For CRISPR, it's going to create a bimodal distribution, right? Let's say 100% of the alleles are going to be modified. Some of these, we think that at least 60% of them, in which you introduce a frameshift mutation, are going to knock out the protein. 30% will not, maybe. So you'll see a bimodal distribution. If you're going to look at this using Western blots, the amount of depletion could be very similar for shRNAs and CRISPRs. But if you would look at the single cell distribution, then it will be very, very different. And what we think is that those cells that are actually completely depleted out of NF2 in this case, those are the cells that actually give you this very strong sensitivity. And that of course is going to depend on the specific gene and the specific cellular model, whether a complete knockout is actually really important. But we think that this is the case here. And of course, the genomic modification in these cells is actually also, when we quantify the percent of indels just by PCRing NF2 and seeing how many cells actually have a modification, then it's almost 100%. Yeah, 100%. OK, we validated additional genes and did a side-by-side comparison with shRNAs. You see here NF1, you see that it was found, indeed was found in the shRNA screen. And we also found it with CRISPRs. MED12 actually gives an effect in shRNAs, but probably still it was not. It was found in a different system, not in a pooled screen. Something interesting. So at this point, a lot of people, when we look at the GFP histogram that I showed you initially, then a lot of people ask, OK, how come you get 100% knockout? Like you would expect actually that 30% will give you an in-frame mutation, right? And the reason for that is, we think, is that when we look at the distribution of indel sizes for different targets, you see that the distribution is very, very wide. It's not like you see 2 base pairs, 3 base pairs. You can see deletions of up to like 30 base pairs, 20 base pairs, like very large deletions. And very large deletions, you would expect that even if they are indeed in-frame indels, they would still abolish the protein function. And one interesting observation here is that this distribution is very, very different in different loci due to some reason that we don't really know. But it's something to think about. So we looked a little bit into our targets from the screen. So this is the MAP kinase pathway. And a mutant BRAF basically keeps this pathway hyperactivated. Okay? And the drug is designed to target the specific mutation in order to shut this pathway down. So we looked into our hits to see whether they actually correspond to something interesting. And indeed they do. So NF1 was shown to be a repressor of BRAF. So it actually makes sense that if you knock it out, you get an increased expression of BRAF. COL3 is an E3 ligase. And it's actually counterintuitive in a way, if you think about it. But it was shown to have a direct interaction with NF1. So it's very interesting that we actually found COL3. Other genes are also shown to have an effect. So MED12 is an inhibitor of TGF-beta. NF2 is a very interesting example because it actually doesn't, at least with the current knowledge, it's not expected to act in the MAP kinase pathway. And we think that it actually acts in parallel pathways. Interestingly, these 3 hits are all part. These are also these are transcription mediators, and they're all part of the same complex that actually enhance c-Myc expression. So this was also very encouraging because all of these genes that we got completely independently were part of the same complex. After the paper came out, we were flooded by requests, and were not fast enough to give the plasmids to add genes. So So we basically were very busy packing packages. You see here, Patrick here, packing some stuff. And yeah, next. So we sensed that we also improved the system quite a bit. So we made improved libraries based on more advanced information on off-targets. We improved, one of the most important things is that we improved the lentiviral delivery system. So delivering Cas9 is kind of difficult because Cas9 is so big. And so you basically get very low titers when you try to make virus with Cas9. So this was done mainly by Neville, my collaborator, and he managed to clone some stuff back and forth and managed to boost the titer of the one vector system quite a bit. So this is really great because it enables screening in primary cells and more difficult systems than cell lines. Yeah, I'll just sum up quick. So I showed you that lentiviral delivery of Cas9 is extremely effective, can reach 100% modification. We get much higher consistency in the screen compared to sgRNAs. We found new and established PLS resistance mechanisms, and we also validate our results. And I think one of the most interesting things here is that when you compare CRISPR to sgRNAs, in sgRNAs you're limited to the transcribed space, right? And in CRISPRs you can target whatever you want in the genome, almost whatever you want. So when you think about these, using custom libraries of sgRNAs to target different types of different regions in the genome, you can really screen for a lot of things. You can screen for enhancer elements that are responsible for a specific phenotype. So once you have a system, you can really use this to explore all sorts of different places in the genome. And it's very exciting because, you know, a lot of systems or genomics people have been worked very hard to read out things from the genome, like reading out epigenetics, reading out transcription. And now we can actually perturb the genome in a high throughput way. And that's, I think it's a very, very exciting phase in biology generally. Okay, I'd like to thank the Zhang Lab. Neville is my co-author on this paper, and we worked very closely and still together. Fung, other people, the RNAi platform and the broad funding. And I'll take this opportunity also to thank, on behalf of the invited speakers, to thank Corey and people that invited us over. So it's been a very pleasant, short stay. But thanks for that, and thanks for listening. If you have any questions.